Okay, so today we're going to be talking about the spread of signaling throughout cardiac muscle cells. So to start, we have a few key layers we want to make sure we take note of. So the first is these intercalated discs, and these are just important because they kind of, hold these cells together. We then have our gap junction.
And the thing about our gap junctions here is these allow charged ions to move across.
And then we have our desmosomes. So as we move down here, we'll be able to look at kind of the flow of our cardiac conduction system. So it's important to note first that a node is just a group of myocytes. So I'll write that up here. Node equals group of myocytes. So first, we start at this sinoatrial node. And so this is going to be autorhythmic, which just means that it generates its own action potential. You may have even heard of this being called like the pacemaker. So it's just important to note that. And so our sinoatrial node, or our SA node, is going to be right here. And so we can see that it's at the top of the right atrium, which makes sense because that's, where our blood is starting to come into the heart. So it makes sense that our action potential would kind of start there. So we move down to our AV node, our atrioventricular node, and this takes place right here. So it's kind of right in between that right atria and right ventricle, which makes sense of the name, again, that atrioventricular node. And so this atrioventricular node is it's like the sole electrical connection between the atria and the ventricles. So it's very important, and it keeps them in pace with each other. So then we move down to the bundle of His, which is right here. It's just kind of a little bundle that helps keep the charge moving, and it allows for the charge to flow down into the ventricles. So then as we move down into the ventricles, we have our bundle branches. And as we move into our bundle branches, this is what they look like. Or that's at least our right bundle branch, I should say. Our left bundle branch is right here. And these allow the spread to, flow or spread around the ventricles. And then lastly, we have our Purkinje fibers, which just kind of allow the charge to really spread into each cell and allow the top ventricles to conduct as well.
So before we move on, I just want to kind of reiterate the pattern that occurs here. So this is just basically the flow of this is that the SA node initiates the heartbeat, and that sends signals that spread across the atria, and it reaches that AV node. That then introduces a crucial 0.1 second or 100 millisecond delay before passing the signal on to the ventricles through the bundle of His. So our node, we can add this down here, introduces delay. And that delay ensures that the atria fully contract and empty the blood into the ventricles before the ventricles start contracting. So ensure all blood is in ventricles before the contraction. And then, so this ultimately prevents backflow. And that kind of optimizes that cardiac efficiency.
Down here, we can see that our kind of another view of like the charge spreading throughout. So we can see that it's highlighted right here, our SA node. It spreads, it goes down to this AV node, and then it spreads down into our bundled branches. So that's kind of important to note. And then also important to note, as we talked about our parts of the cells up in that first slide, that they spread via the gap junctions that allows that, conduction to spread between the atria and ventricles. And then again, just remember that this SA node is that pacemaker. Yeah.